Participants Maya argues for allowing tightly regulated germline editing, permissive but careful. Celine argues for bans, strict moratoria, until stringent conditions are met. Maya, let's separate what's possible from what's wise. On possibility, we're past speculation. We've seen pathogenic variants corrected in human embryos in the lab, and we now have editors that minimize double-strand breaks. Prime and base editors aim to write precisely, not slash. And mitochondrial donation shows that heritable interventions can be integrated under strict oversight. If parents face a near-certain risk of passing on a devastating mutation that screening can't avoid, shouldn't we build a pathway for a carefully governed option? Celine, a pathway might exist one day, but today's road is full of holes. Embryo editing still produces mosaic embryos at non-trivial rates. We've also seen large, unexpected deletions and complex rearrangements right at the on-target site. Those are not minor glitches. They threaten genomic integrity in the very cells that would become a child, and every descendant. Until those failure modes are rare, predictable, and detectable at scale, carefully governed option is a euphemism for unacceptable risk. Maya, that's why we invest in getting the failure modes down. Editing earlier in development, improving pegRNA design, transiently modulating repair pathways. These are active research fronts. We can combine them with deep sequencing, long-read methods, and multi-omics screens to catch aberrations. Science doesn't jump from version 1.0 to clinical use overnight, but the trajectory is moving toward precision. A regulated pilot confined to monogenic diseases with no alternative could be safer than you think. Celine. Pilot sounds responsible until you remember that germline edits don't end with one child. They propagate into a family line. If an undetected structural variant increases cancer risk at 40, you find out decades later, possibly after it's already heritable. Somatic therapies let us correct mistakes in an individual. Germline edits make errors permanent. That's a categorical difference, and it demands a higher threshold than clever lab metrics. Maya, I don't dispute the higher threshold. I'm saying we should design a path to reach it, one that includes multi-generational animal studies, shared benchmarking datasets, independent replication of preclinical pipelines, and public registries with lifetime follow-up. We won't meet those thresholds by banning research or declaring the entire area off-limits indefinitely. Meanwhile, families with lethal mitochondrial variants are already benefiting from regulated reproductive technologies. That should be our model: strict indications, expert oversight, and transparency. Celine, mitochondrial donation is instructive mostly because it shows how narrow and supervised such pathways must be. Even there, authorities moved carefully, case by case, with long-term monitoring. And that's targeting the mitochondrial genome, not editing nuclear DNA with its infinitely larger landscape of risks. For nuclear germline edits, the list of unknowns is still too long. Unforeseen off-target effects, epigenetic consequences, developmental impacts, and gene-environment interactions we barely understand. Maya, but the status quo is not benign. Pre-implantation genetic testing helps when good embryos exist, but some couples carry variants that make select a healthy embryo a fantasy. Others face repeated IVF failure. Adoption and donor gametes are options, but they are not substitutes for many families. A blanket ban tells them, your line ends or you accept the risk. A regulated exception says, we will try to make it safe and we'll be honest about what we know and don't know. Celine. Honesty also means acknowledging societal consequences. Once a clinical door opens, even a narrow one, market pressure pushes it wider, from avoid lethal disease to reduce risk to enhance. Regulation can slow that slope, not eliminate it. In a world with uneven access, the benefits accrue to the wealthy first and the harms land on the vulnerable. We need legitimacy from society, not just from a committee of experts. That means broad public deliberation, not technical sign-offs. Maya. Agreed on legitimacy. Public input, citizen assemblies, and transparent criteria should set the boundaries. But fear of hypothetical drift shouldn't paralyze progress on genuine disease prevention. We draw lines in other sensitive areas: organ donation, reproductive medicine, end-of-life care, and we enforce them. It's work, but it's doable. Celine. The analogy breaks because germline edits rewrite future people who cannot consent, with risks that current methods cannot fully measure. Informed consent is already complicated in reproductive medicine. Here, the subject is not even born. That alone should tilt us toward extreme caution. If we can prevent disease by screening or by somatic therapies later in life, those should dominate. The case for germline editing should be extraordinary, not routine. Maya. Then let's define extraordinary crisply. Severe, highly penetrant, single-gene conditions with no feasible embryo selection path, validated editing pipelines with independent audits, comprehensive genomic integrity checks, and lifelong registries with healthcare support. I don't advocate routine use. I advocate building the capability responsibly for those edge cases where nothing else works. Celine. And I argue we aren't there yet. The technical metrics you cite are improving, but the distance from controlled lab embryos to healthy adults with children is vast. Moreover, global governance is fragmented. A permissive clinic in one jurisdiction can undermine cautious standards elsewhere, and families will be tempted to travel. Before contemplating clinical use, we need enforceable international norms, not just local approvals. Maya. International harmonization would help, but waiting for perfect consensus is another kind of paralysis. Realistically, a small number of trusted jurisdictions could establish gold standard protocols and share data under international observation. That can seed common norms. Meanwhile, a research ban would drive activity underground, which is the least safe outcome. Celine. I'm not proposing a research ban. I'm proposing a firm clinical boundary while research proceeds. No embryo transfers after nuclear DNA editing. Advance the science, publish the failures, harden the detection methods, compare editors, and build oversight. If the evidence clears a high bar and the public agrees, revisit the boundary. That stance protects potential children and maintains public trust. Maya. Then we are closer than it seems. We both want high bars and public legitimacy. I'm focused on designing a narrow clinical path for the rare cases with no alternatives. You're focused on ensuring that the science and governance truly deserve that trust before any clinical use. Perhaps the compromise is a staged framework with explicit gates. Once predefined integrity, safety, and consent criteria are independently met, limited clinical trials begin under international supervision. Celine. A staged framework with immutable gates and automatic shutdown if red flags appear would be the minimum I'd consider. Until then, bans or moratoria on clinical germline editing protect real people from irreversible mistakes. Maya. And until then, let's invest in the work that will actually answer the open questions so that future decisions are driven by evidence rather than anxieties, however understandable those anxieties are. Celine. On that we agree. Evidence first. Humility always.